Okay, so our last speaker is Seth Winfree. Seth is at the Indiana University School of Medicine and is part of the Indiana Center for Biological Microscopy at IUPUI. He has lots of expertise in large-scale data analysis and correlating multiple technologies, and today he's going to talk a bit about what he's doing as part of KPMP, and the title of his talk is Bi-Directional Interrogation of Image Volumes and Segmented Cells with 3D Tissue Cytometry.
Good afternoon. So thank you to everyone here for setting up the workshop, and thank you to Sanjay Jain for specifically inviting us as well. I'm sort of filling in for my mentor, Tarek Ashkar, who's one of the PIs, part of the KPMP program. And today I'm going to talk about some technology that we developed in the lab to go after our own work, but then are also applying to the KPMP as well. So as we're sort of implicitly in the KPMP focused on is moving from this space where we have one-size-fits-all medicine towards precision medicine. And really that's important because we've moved through the process of stratified medicine, but really we're beginning to appreciate how important individuals are in the actual clinical course that individuals are taking. As part of the KPMP, we're trying to transition from really stratified medicine to precision medicine. We're addressing the needs of individual patients based on molecular diagnostics. So, and of course, that's coming from a one-size-fits-all or stratified medicine approach. So, there are a couple ways that we can think about that is that at the end of the day, to make this achievable, we're really going to be looking for very small changes that can have relatively large effects. And those changes really are going to be, to use some imagery, a needle in a haystack. And that really is going to invoke the requirement for the application of high-resolution methods with high sensitivity. And really, in the end, because we don't really know what those needles are going to be, we're really going to need to be operating in a multi-parametric data sets and have the ability to analyze and interact with those kinds of more complex data. And at the end of the day, I think the last point is we'll be looking for what we think will be small changes that are unique for a given patient and for a given disease. So what our tissue interrogation site is doing as a part of the KPMP is doing 3D imaging. And I think I don't need to make the argument for the HubMat community, but we often have to make the argument for why 3D is important. So here on the left-hand side, I have a single optical section, confocal, stained for 3 different things: nuclei, resident macrophages, and filamentous actin. And you can't really appreciate the degree to which the MHC2 positive cells, those are the red ones in both panes, are really wrapping around the tubular structures as well as the glomeruli. So, and of course, as Simon Watkins just very elegantly showed us, the complexity of tissue is not, of course, at that small level. We're at much larger scales. We're at mesoscales. And even the kinds of tissues that we'll be analyzing and collecting in the KPMP are really on this scale. We're looking at up to 1 centimeter in length or more and 1 millimeter or so in diameter. So this is just a higher resolution. The quality is a little blown out on our particular view here, but you can get an idea of what the resolution is. We really have cellular resolution across multiple markers. There goes a glomerulus. We'll zoom back out here. For the sake of time, I'll keep moving us forward.
It's beautiful.
So what we set out to do was, if that's the scale of the data, and that's the level of resolution that we can capture, how do we actually get it, quantifying it in some sort of meaningful way? And we sort of took our inspiration from flow cytometry. And so in flow cytometry, the basic approach is, instead of leaving the tissue intact, we take the tissue out of its context. So let's just pretend it's like a mouse tissue, for instance. We grind it up, develop single cells, stain them in some specific way, run them on the flow cytometer, and get an idea of what kind of cell they are based on their surface markers, based on the stainings that we use. So we figured we could probably take a similar approach, but leave the tissue intact. So, and that really embodies the idea of tissue cytometry. So to achieve this goal, we really have to solve one really basic problem. What is the most basic unit that we care about in the tissue itself? And for us, we decided the best fiduciary for a given cell is going to be the nucleus. So in all of our approaches we take of 3D cytometry, we're really counting individual cells or assessing individual cells based on their nuclei. So our segmentation approach can vary. There is no perfect solution for all cases, at least it hasn't been developed as of yet, but here's just schematically what you can imagine. So if we have a piece of cortex on the left, we can isolate the nuclear channel, and then we can do some level of segmentation in 3D. Then to continue the process, once we've actually counted all of those cells, for instance, we can associate that with signal that's in the tissue, so looking back at the image data in 3D, and then classify and count it. So there are a lot of strategies for achieving this. We wanted to develop a platform that really integrated the entire process in terms of the imaging processing. And we wanted to be able to do the image processing, which is required for managing inconsistencies in the imaging platforms themselves. We wanted to have places for adding in segmentation as well as ways to do analysis. And the way that we think of this is that really there's two levels. There's tool use and then also development and growth for this actual implementation. So what did we come up with? This is our nascent tool that we've been using both in the KPMP and in our own research. And really the basic concept is we're taking, again, those 3D images, passing them into a pipeline, our workflow process that allows us to do image processing, segmentation, and then presentation of the data in a flow cytometry kind of way. So basically an XY scatter plot. We then can take that XY scatter plot, where every spot represents a segmented cell, ask questions about the intensities in the other channels, so essentially associating a signal with those cells, and then determine where they are back in the original image and also characterize them. So for instance, in this case, we're going back and looking at the green channel and identifying where those cells are, and also getting basic statistics on them, how many there are, as well as a red overlay to give you an idea of where they are in the specimen itself. So I'll go into a little bit more detail in the next slide. This is that same volume that was in the flow there. And basically, if you're looking at mapping just DAPI intensity, so just the intensity of the nuclei themselves, versus this marker aquaporin 2, which labels a specific subsegment of the nephron, we can go through and do cytometry on and identify, for instance, 9.5% of them in this particular field are positive for that particular marker. We can get an idea of where they are in panel C, so that's the red overlay. And then we also, once we go back to the image itself, have the ability to draw ROIs in the image space and actually highlight them back in the analytical space. So this is a basic approach that we've applied to a number of cases. This was in the initial publication, where we just correlate the degree of infiltrates with clinical outcome, or well, clinical markers, I'll say. This again was just these two particular patients, so I wouldn't draw any specific conclusions from it, but it shows that we can and could have the possibility of associating cytometric measures to clinical measures. So of course, what I've just mentioned here is a 4-channel space, and although that's usually or traditionally the maximum in the confocal world, we have delusions of grandeur and would like to have more channels and more information. So we would both like to, for instance, understand structural components as well as infiltrates, so immune cells, for instance. So we've developed a strategy to get us 8 channels into these large 3D volumes. This is, I would say, in the grand scheme of multiplexing, as much as we've attempted, there are other approaches that deal with non-3D structures that allow you to multiplex to 32, 64. But again, because of our interest in 3D, this is where we've focused our efforts. So then that starts to really complicate the analytic space, because now we're going to have, for instance, in this case, this particular piece of tissue, 150,000 nuclei, and if we're just looking at intensities, there could be up to like 70 intensities and morphology, somewhere around 70 attributes for each individual cell. And those are pretty, in the grand scheme of things, rudimentary attributes. They're not, I wouldn't call them necessarily complex. They're basic ideas about what the shapes are or what the intensities are, and to some degree what the distributions of those intensities are around those nuclei. So this is where we've invoked an additional model where we're actually incorporating both that supervised analysis, which I covered, as well as more of an unsupervised analysis. So then we can really take these higher dimensional spaces, higher dimensional data, pass it through our same pipeline, do both supervised analyses and some level of unsupervised analysis, but still have the ability to link it back to the biology in situ. And I think that's an important distinction because there are essentially toolkits and APIs that exist to allow you to do different aspects of this process, but this is the intent here was actually unified into one thing so that you can go from what are the adjustments that we can do in terms of image processing, changes we can do in terms of segmentation, and what sort of consequences that has in terms of the analysis on the other end. And then we can go backwards. We can go back all the way to the image processing because we realize we've done something inappropriately or less effectively, and then go all the way back to the analysis as well. And that gives us a sort of bi-directional access to both the original raw data and the analytic output. So this is the incarnation right now, and I won't go through the whole thing. I was going to do a short video demonstration. Yep. And just to go through the process, because it'll be a little bit more informative than me just talking about it.
There you go. Oh, that was,
Oops.
no, it must be because it's in the video screen. It's this one here.
Yeah, there it is.
It's not.
Can you minimize the PowerPoint?
Let's see here.
There we go.
Just share the reshare the.
We'll reshare the other.
Yeah, share your window, not the desktop. Hold on just a second.
There we go. There we
So
go.
basically, in the video, what we will see is that basic process occur, and we'll cut it short for the sake of time. So I don't want to.
Do you see the right?
Okay.
Don't see the video.
Yeah, we're
Trying to find it.
We're trying to pull it up here.
Maybe in the downloads directory, maybe it's
It was already open.
It was
I see
open.
the case
There it is. It's got a
Videos.
white box on it.
Yeah, right there, Windows Media Player, upper.
Up top, right there.
Show all windows, bottom right. Bottom right. Show all windows.
Scroll.
Oh, there it is, right there.
There it is.
You can cut this out, nobody else will have to know.
All right.
That's all right. Okay, so let's go ahead and just press play. Or, yes. All right. So here, this is that same volume again. I left it in here for consistency's sake. And basically, we have two tubular markers. Everyone seeing it? Yes, you're seeing it. Okay.
Yeah.
So there are two tubular markers. One is sort of a magenta purplish color, and one is a yellow color, and there's also green for F-actin. And here I've already segmented the cells. I'm going through and doing the gating on the scatter plot itself, and then trying different kinds of channels, and then or a lookup table. So here I've applied a hierarchical clustering to the original data set, so the intensity measures, to try and pull out subpopulations. And here's just doing it with dimensionality reduction to get a better separation of that space. And then associating intensity right there, pull out just the F-actin of the green positive cells, so those ones that are highlighted on the left-hand side. And then lastly, I'll apply the hierarchical clustering that I've already done to show that that population, although by intensity looks like it's one population, by clustering it clearly pulls out as two populations, and then actually mapping that. And then in addition to that, mapping it back to the original volume as two different overlays, just to get a feel for what the different populations look like. Basically, a subset of cells that are in the glom, two different subsets in the glomerulus. So you go ahead and back to the presentation.
Which means we're going to have to bounce again.
We're going to bounce again. So then, it's still playing in the background. Making it work.
The problem is the control. Every time I try to move the controls and stuff like that, nothing's happening.
Is that you develop it to work within Fiji? It's like
Yes. So it all works in VG.
So what slide were you on? Was it this one?
So we're on the one down the middle and the bottom. 17.
Okay.
So importantly, I just had a question from the room here about where the program operates, and it was developed as a plugin for Fiji, and everything you just saw wasn't done on some sort of supercomputer that's done on a MacBook from 2-3 years ago. So in terms of the heaviness of it, it's pretty lightweight program and fairly effective in its use. So what can we do once we have this kind of imaging data? And this is where we get more into vision than into actual product. What we kind of imagine is that we can be generating data in the imaging space. So for instance, here we're looking at cellularity of glomeruli. But there are two levels at which this operates. This operates both at the level of the we're having technical difficulties with everything.
Yep.
Here.
Nothing's happening. With
Nothing's happening with either.
No.
We've gone into.
I think we've hit. Okay, we're going to stop sharing for a moment here. So this is yours. I'm going to close out. Connie, that's yours,
Yeah,
right?
sure.
So let's close this one here. And this
You
is
have
yours.
two of mine over there. There we go. I was going. All right. Take us back. There we go.
Okay.
So the imaging data that we're collecting.
You just share again.
Go share.
No.
Yeah, sure.
That's a little-
No.
Yep.
Okay.
All right, so everything I've talked about up to now has been at the single cell level and characterizing the individual cells. But the imaging, of course, also operates at the next level of complexity in terms of different subsegments of the nephron. So it's very natural for the imaging to function as kind of an anchor point between stuff that is at the level of the glomerulus, for instance, so where we might have resolution in terms of proteomics, different kinds of laser microdissection-based omics, as well as metabolomics. But of course, because it's cellular, still not doing it. But of course, because it's cellular, we also can connect it to single cell RNA-seq and other single cell approaches.
Yeah.
Totally kill it.
I'm gonna-
4 more slides. Do you want to try? There's a hub map. Go exit out and over on the right. More. Just a different one right there. You have 2 open or something?
Yeah, that one makes sense.
Yeah, I think you might have been grabbing someone
Well, the
else's. Yeah. Let's go in there and
Just the
then.
closest
Yeah.
one.
Close this one. I don't think that's the one we should have closed.
I don't think that was one we should have closed. No.
Yeah. So,
That's all right. Just open it over here and we'll
yep.
zip ahead. Apologies for the technical. Thought we were ready based on our dry runs.
Well, this is being run on a Nook, so it may have reached its mental capacity. All right, so let's enable editing.
17.
Wait for it to draw. Okay, 317. Full screen.
Yep. All right.
Nope, we got to share it.
Oh, yep. Sorry, getting ahead of myself.
Yep. Okay. And
Yep.
share. Is it
All right.
working?
Yeah.
Still not working.
No. I don't know what.
I think what before.
I'm
There.
working. I'm
There.
working.
Oh, you're working.
There.
All right, let's get rid of this guy. All right. Okay. Where were we? All right. So what I just showed you was the demonstration. Whoa.
You can just click. You can just click on the screen.
Yeah. So I just showed you a demonstration about how we can quantitate individual cells, classify them. Because it is an imaging paradigm, we can also, of course, think of things in terms of structures at the level of the nephron, so essentially subsegments. So in this case, we're looking at the glomerulus and looking at its cellularity. Now that, of course, means that we have two different levels of resolution. We both have levels of resolution that are compatible with these kinds of techniques, for instance, proteomics, metabolomics, and different varieties of laser microdissection. But because it's also a single cell approach, and we can map into analogous kinds of spaces, we have the potential to map between both the single cell RNA-seq data as well as the imaging data if we can find the correct correlates. So if then we can achieve that, that's one of our active areas of work with the central hub Currently, what we can imagine or what we envision is the, there it goes, the ability to go through and identify specific structures in the tissue and associate those structures with, for instance, differential expression based on the transcriptomics, metabolomics, as well as subsegment-based LMD transcriptomics. Really to give you a hook into the molecular data in the imaging data. So, of course, we can also essentially go the opposite direction where if we have clouds or clusters in the single cell, single nuclear RNA-seq data, we can correlate that back with the imaging data. And for instance, if we pull out just an individual cluster in the imaging space, we can associate that in the tissue itself and pull out specific specific locations where we see those infiltrates, for instance. So that's two ways in which we envision the process occurring in terms of how we could go from the imaging space to the omics space. So in the end, the other way that we see the data being used in the long run is really making a model across a variety of scales. And really this is how do we map from individuals and the patient biospecimens and the data we collect there to the outcome or to an actual model. And we imagine incorporating the data that we're collecting in the KPMP as well as the sparse data, which is essentially sparse data because again we can't get entire structures in all cases, to really build that average model for the kidney, both across pathologies and also as in terms of reference. So this is the approach, and we're in the nascent stages. I've started this project in the lab where we are taking data from the KPMP project, the sparse 50 micron section 3D data, combining that with reference 3D volumes that we've collected based on after clearing the tissue, as Simon Watkins just mentioned. We're using a similar approach, and then using a number of deep learning strategies, trying to figure out the best way or the most representative way to take that sparse data to build up those specific models. Again, we envision that, at least here in the beginning, being a reference, but as we move in the KPMP program to either AKI or CKD, also developing models for those particular pathologies, disease states. All right, so in conclusion, there's a lot of them there. We can interrogate at high resolution our 3D fluorescence imaging data sets, and that's both resolution in terms of the spectral space as well as the physical space. We have tools that we're developing that allow us to make that occur. We have ways, or we have shown that there's links between pathobiology in situ and how we could possibly use that for precision medicine in the future. And we also have a clear path in terms of multivariate analyses to mine these large, complex data sets that aren't as easy to approach on first blush. And I think through the work with the KPMP, we're at the stage where we're developing strategies to actually integrate the imaging data with our omics data to further expand the signature of disease state as well as the signature as reference state of normal, quote unquote. And then hopefully by the end, I think one of the positive outcomes of the imaging data, I think will give us in the long run a model average kidney, at least in the tissue that we're sampling, even though we're collecting only sparse human biopsy data. I think with conclusion, yes, is thanking everyone at IU that's associated with the KPMP project. So Ashkar, Dagger, and Eden are the PIs. The rest of the group that's helped both on the transcriptomics as well as the imaging side. And then all the funding. Thank you.